All right, I think we'll get started. It is my absolute pleasure to be chairing this session entitled Diabetes and Glucose Metabolism. We have a diverse lineup of speakers, all with expertise in glucose metabolism and muscle metabolism. I'll be your first presenter, Dr. Michael Riddell from York University in Toronto, Canada. And then my presentation will be followed up by Dr. Nair from Rochester, and then Dr. Sylow from Copenhagen, and then finally Dr. Cartee. And I'll be giving a little bit of an introduction about how privileged we are to have these speakers today when they go ahead and take the stage. But I thought for my introduction, who better to give that than maybe Tom Hawk. Are you here, Tom? Just kidding. I'm sure you'll say something nice in the question period. But I'll be giving a little bit more background about who I am and my research interest as I go through this first lecture. The title of my talk is on the glycemic responses to exercise in type 1 diabetes, lessons learned from the T1 Diabetes Exercise Initiative, or DEXI for short. And thank you, Dave, for asking me to be a part of this symposium, for chairing this session. So in the next 25 minutes or so, I'm going to give an overview of what type 1 diabetes is and how it's currently managed and how the evolution of management has really changed in the last 100 years. I'm going to talk next about the glycemic trends that we see with exercise in kids and adults who have type 1 diabetes. And I'm going to talk about this more recent study that we've just completed called the T1 DEXI study. I'll tell you a bit more about the study purpose, how we underwent the study recruitment during COVID, and some of our results to date from this fairly large cohort of a real-world study of exercise in type 1. So for those of you who don't know, type 1 diabetes is an autoimmune disease, and it's highly selective to T-cell infiltration of the insulin-secreting beta cells. If you look at a pancreatic islet slice in a cadaver dissection, let's say of a young child who's died from some other cause, you'll see T-cell infiltration in the islets, as shown here, but specifically it seems to be focused on the beta cells of the islet that produce insulin. Insulin is stained pink in this slide, and glucagon is stained blue. And you can see these T-cells infiltrating the islet in this brown color. Now, 100 years ago, this year, insulin was first used therapeutically in Toronto. This was a Nobel Prize-winning discovery in Toronto that I'm quite fortunate to shed the benefits of as someone who's living with type 1 himself. Now, 100 years ago, this is kind of the phenotype of someone diagnosed with type 1 diabetes. This is Teddy Ryder, who was the first patient in Toronto to receive insulin by injection. And you can see this cachexic figure of what hypoinsulinemia does to a child with type 1 diabetes. And it's actually pretty transformative what happens 6 months later after Teddy was receiving his daily insulin injections. Almost a restored, completely normal phenotype. Lucky Teddy, if he was born a year earlier, he wouldn't have survived. And Dr. Nair is going to talk a little bit probably about the benefits of insulin therapy on limiting the catabolic actions in muscle, and we'll learn probably more about how there's dysregulation in glucose metabolism for me, even after insulin therapy is initiated. Now, I come from this topic pretty close to home. I was diagnosed at the age of 14 as an active teenager who wasn't really doing all that well academically, but loved sports. And that diagnosis for me gave me a passion to become a better student and try to become a better athlete, which I never really was. But then it also hit home again when my son was diagnosed. You see, because this is a genetic disease that we pass on to our offspring with this risk of an autoimmune destruction of those same beta cells. It's not purely genetic. There must be an environmental trigger, and we don't know what that trigger is. But the risk for Sam was certainly much higher than the risk for you if you don't have a family member living with type 1. Now, because of this diagnosis, I went to graduate school. I first did an undergrad at University of Guelph, have fond memories of studying metabolism from Lawrence Spreet and Terry Graham. And then I was fortunate enough to go to McMaster University for my master's and PhD, where I worked in the Children's Exercise Nutrition Center. and was able to use stable isotope tracers to look at metabolism in young kids who were diagnosed with type 1 at about the age I was diagnosed, learning at how substrate metabolism was not normalized by insulin therapy, unfortunately. I then went to the University of Toronto and got to work with Dr. Mladen Vranek, who was actually a postdoc of Charlie Best's, who discovered insulin. So that lineage, for me, again, hits quite close to home. And I'm now very proud to have a lab at York University where my graduate students are focusing on the metabolism of diabetes, whether it's hypoglycemia, hyperglycemia, exercise physiology, or what have you. These students have some connection, I think, to diabetes, either because of an interest, a family member, or my excitement and enthusiasm, but what still needs to be done in this space. The technology has changed since I was diagnosed with type 1 diabetes in the early 1980s. Of course, the technology has changed even since Sam was diagnosed just 4 years ago. This technology moves rapidly, and it moves to wearables, insulin infusion devices, ways that you can communicate your glucose level to your mother when you're at school or at university. Sam's at Guelph now, so we can follow his glucose while he's in his first year at university. And that's incredible to me, that the technology keeps improving. But glucose control is still not optimal in type 1 diabetes. 3/4 of patients Sam's age do not meet the required targets to prevent complications from the disease: retinopathy, nephropathy, neuropathy, heart attack. So we know we can still do better. And of course, you'll hear from Dr. Hawk that there's this myopathy associated with the disease, and Tom really coined the term diabetic myopathy. And I love Tom like a brother, like a younger brother. But sometimes I get upset with my younger brother when he says, I'm also deficient in the ability to grow skeletal muscle mass, to have normal mitochondrial density, and to perform sports and exercise. Yet still, there's tons of examples of Olympians with type 1. And there's bodybuilders with type 1, and there's even models with type 1 that now show off the technology required to keep glucose in control as best we can, despite the abnormal physiology we've inherited. Does anyone remember Kate Moss, by the way? Dave, Kate Moss? This is Kate Moss's daughter. She has type 1. Now, I've never modeled for type 1 diabetes in technology, but I thought I'd share a picture with you. This is a picture of me wearing a continuous glucose monitor in around 2003, I think it was. This is the first glucose monitor that could be worn by a patient living with type 1 diabetes to monitor his or her glucose in real time. But look at this technology. It's a wire attached to a receiver, and the receiver doesn't show the data. I won't tell you where the wire goes to. You can use your imagination. But you're wearing an implant connected to a wire to a receiver that shows no data until you take it to Dr. Nair. He downloads it and then says your glucose was shit last week. What good is that technology? Because last week's gone. The technology has changed. Now we know we can wear glucose sensors in small, discrete parts of the body. They can broadcast that glucose data in real time to a mobile device or receiver, and that data can sit in the cloud in real time so parents, caregivers can watch the glucose and nag you when your glucose is out of control, of course. If you're interested more in CGM technology, we have a poster on the use of CGM technology in healthy, elite-level athletes, not diabetics. That's poster 70. And I'd appreciate if we could get some attention there so I could send some pictures back to the company that makes this technology, which is Super Sapiens. This technology does help. I can now see my glucose in real time on a Garmin watch. I can show it on an Android phone. I can correlate it with my heart rate when I go for a run. I can get alerts and alarms if my glucose is too high or too low. I even have an experimental app on my iPhone, not yet commercially available, that adjusts the insulin infusion based on my own personal insulin sensitivity, the macronutrient I've had in the last meal, and whether I'm exercising or not. You'd think this disease was solved. You'd think you wouldn't need a transplant anymore, but it's not. It's still challenging. I now know to avoid some foods like white bread. You can see that schematic there where my glucose was in the normal range at around 5 millimole. Have one slice of white bread bolus insulin when I eat it, and glucose goes to 17 millimolar. I did everything right. Let's talk about exercise. Exercise is critically important for the health and well-being of that person living with type 1. We know all the reasons in the general public why it's important for mental health and physical health. But for type 1, this deficiency that we continue to see in the work by Dr. Hawk and others can be improved, can be normalized if patients are physically active. We know that activity enhances capillarization of skeletal muscle, the biggest sink for glucose disposal when that kid takes insulin and glucose is high. We know it improves muscle mass and bone health, which can be impacted by the disease. It increases insulin sensitivity, and many of the people with type 1 are insulin resistant and overweight now with the advent of insulin pumps. It improves the social aspects of it and well-being. It certainly lowers the cardiovascular risk profile. There's evidence now it lowers the risk for kidney failure, which is exciting. And some of our work suggests it may improve your overall time in glycemic target range, but it also increases the risk for hypoglycemia or low blood sugar. That's the problem that we're focusing on right now from several different lenses. Now, exercise, we know, increases glucose disposal. This is a really neat image visually that shows us that the muscles that are moving are taking up glucose at a much, much higher rate compared to the muscles that are not moving. This is a cross-country ski PET scan that shows glucose uptake into all the major muscles in the upper body and legs, and also into the CNS. And much of this glucose uptake is insulin independent, isn't it? We now know from all the great research presented by people who are at this conference, in fact. That increase in insulin sensitivity and glucose disposal is really good for type 2. But sometimes it's not so good for type 1. Why? Because glucose drops too quickly in circulation and the patient develops low blood sugar or hypoglycemia. I've been so inspired by the work of Eric Richter and Mark Hargraves that I actually start to think about the pathways that glucose disposal is messing up my glucose. And we now know that there's insulin signaling mechanisms that cause my glucose to drop. When I exercise, my insulin goes up in circulation, not down, because of increased blood flow and absorption from where I eject it. We also know that the glycogen content in our skeletal muscles, if we exercise for a long time, seems to be somehow pulling that glucose in after I go to bed, when I fall asleep, and I can have a low blood sugar reaction then. Sure, the mechanisms are interesting, and I'm glad that others like Leakey are studying this, but I'm interested in the clinical prevention of hypoglycemia, and that's what we're going to talk about next. We were really the first to show that when a subject with type 1 diabetes does aerobic exercise using CGM, they get two powerful hypoglycemic events, one during the exercise and one about 6 to 11 hours later when they're sleeping. And this duration of hypoglycemia, glucose less than 4.9, is life-threatening. It's also life-threatening if you're driving a car, you don't know which lane to be in. It's also life-threatening if you're on a run with Tom Hawk and you don't have any carbohydrate in your pocket. And Tom has saved me on numerous occasions. But he's not there when I'm at bed, sleeping. That's mostly my wife's job. Mostly my wife, unless we're at a conference, and it's 1992. Remember, CGM was originally a blinded device. The patient couldn't see the glucose. And this is a very sad story of someone living alone with type 1 diabetes wearing a glucose monitor for the sheer purpose of showing the data to his physician at the next clinic visit. He'd done evening exercise after a carbohydrate-rich meal. The evening exercise, of course, brought his blood glucose down, but he didn't develop hypoglycemia. The carbohydrate-rich meal brought his glucose up, but then that insulin sensitizing effect in recovery causes glucose to drop to fatal levels. He was found dead in bed with a vitreous glucose of somewhere around 20 milligrams per deciliter or less than 1 millimole. Exercise can kill this patient population, and it does kill it. And the insulin pumps are good, right? Like they'll deliver insulin at a physiologic rate based on the foods that you eat, but the early generation insulin pumps, although they would alarm that a child glucose is too low, that kid may not wake up, like in this particular case study. where the kid slept through the alarm. In fact, the kid had a seizure and almost died after 2.5 hours of mild hypoglycemia caused by being at a camp. And look, the insulin infusion actually went up because of the dawn phenomenon. Typically, kids are more insulin resistant in the morning. So pre-programmed into the pump was a rise in insulin delivery, even though this kid was at life-threatening hypoglycemia. The newer pumps are getting a bit better. Based on the research that I've published and others in this field, they now can prevent nocturnal hypoglycemia. They have what's called a low glucose suspend function, that as the glucose drops towards hypoglycemia, they can minimize the time in hypoglycemia by turning off insulin delivery. In the cohort, the glucose rises when the pump automatically turns off insulin delivery compared to if the insulin pump was like the one case study I showed you before, where there's prolonged hypoglycemia overnight. But the problem is you have to forecast the hypo, because if you turn insulin off, it might take a while for it to drop in circulation. It might take anywhere to an hour. So the newer systems have a predictive model where they forecast low blood sugar in a 30-minute horizon and then reduce the insulin delivery rate. And some studies have shown that this works well after exercise in preventing nocturnal hypoglycemia in most cases, but not all. But these pumps are marketed for being useful for exercise as well. And the glucose turnover during exercise is a lot higher than during rest, isn't it? So do these pumps also work during exercise with these predictive algorithms? Kind of, but not all exercise forms are the same. In fact, some forms of exercise, you actually want higher insulin delivery. Like if you're in a competitive event, mixed martial arts, hurdles, or a basketball game where there's competition stress. And we now know that we have to have different algorithms for different types of sports. And another handcuff with this disease is patient variability. If you take a number of individuals with type 1 and you give them a constant relative aerobic workload, you still see huge variability in the individual glycemic responses. So no one pump is going to work for all individuals. And no one pump is going to work for different forms of exercise where the glucose excursions are in the opposite direction. This becomes very technically challenging to manage. We have published in laboratory, carefully controlled laboratory studies that these smart pumps, automated insulin delivery systems, do improve time in range, and they do reduce the hypoglycemia. But these studies were industry-sponsored and very carefully managed in our hands. What do they look like in the real world? Are they working? That was always in the back of my mind. So that leads us to this project I'm going to finish talking about. That's the T1-DEXY project. We know that there's variability in the individual glucose responses to exercise in type 1, and exercise comes in different forms, and sometimes it goes up and sometimes it goes down. This makes the development of these systems challenging. Many are afraid of exercise because of hypoglycemia, but our knowledge of what causes the variability is actually quite limited. The studies have never been powered to determine if sex matters, if age matters, if BMI status matters, if macronutrient intake matters. They've never been powered that high. And there's really no real-world data on whether these pumps work during exercise. So this study tries to address issues around patient variability. What are things we can measure and assess and predict that'll help better inform these insulin infusion delivery devices to make them exercise smart? Our goal is to develop new systems that know you're exercising, know the type of exercising you're doing, and your own individual patient characteristics that'll predict your change in glucose. And it's an ambitious goal. And you can't do this alone, and you can't do this without funding. This is a major, major undertaking supported by the Leona and Harry Helmsley Charitable Trust. They donate more money to type 1 diabetes than NIH and all the other funding agencies combined, because they have a family member with type 1. This study is run out of the Jabe Center for Health Research out of Tampa, Florida, and I'm fortunate enough to be the study co-chair for this research. So what did this study involve? It was an ambitious study of trying to recruit 600 individuals with type 1 diabetes. This is a disease that's 1 in 1,000. It's not like type 2, which is maybe 1 in 3. have these individuals have wearables on and doing prescribed exercise at home that we carefully designed for them. This is the adult study that's just completed. We're undergoing the pediatric study right now. We targeted relatively active people with type 1 on a range of insulin infusion therapies, including closed-loop systems, open-loop regular pump systems, and regular insulin delivery by either needle or an insulin pen. They did study-designed exercise for a period of about 4 weeks, and they wore body sensors that were donated from Dexcom and Google Verily Health to monitor things like heart rate, body temperature, accelerometry, etc. We also developed a study app for this that allowed us to quantify the food they ingest with food photography analytics, run out of Pennington Biomedical Institute, where dietitians actually do the analysis for us. Not in real time, unfortunately, but at least for the purposes of the study. You can better contextualize exercise with these apps because they can tell you what the intensity was with using an RPE scale, whether it was competition, stress or not, or whether it was habitual activities of daily living. We designed exercise videos actually at York University, and we tailored the exercise to either purely aerobic, high-intensity interval type training, or resistance exercise training. And there's lots of leftover material from the study, like TheraBands that I'm trying to get rid of in my basement, and smartwatches, but you have to maybe slab me a 50 for that. Each exercise video lasted about 35 minutes, so it was not very long exercise, and we successfully recruited 561 adults with type 1 diabetes, randomized to aerobic, mixed, or resistance exercise. We did this pre-pandemic through the pandemic. And who knew that an at-home exercise study was a good idea in 2019? Shit lucky. We had to screen almost 2,000 individuals to meet the randomization requirements. 162 in the aerobic arm, 165 in the HIIT arm, and 170 in the resistance exercise arm. Again, this is not a training study. This is just a 4-week intervention, snapshot, observational study. Overall, the average age was 37 years, ranging from 18 to 70, predominantly female at 73%, and most were meeting their A1C target of less than 7. The average was actually 6.6, which is impressive. They were fairly lean, but we had a good split between those on current closed-loop systems in the United States, those on standard insulin pumps, and those on standard insulin injection by needle, which I'll come back to. They had some level of hypoglycemia awareness. They could feel hypoglycemia during exercise, which made it a bit safer for us to recruit. We had no deaths, thankfully. That could have been a bit of a career killer. What does the data look like as it streams in near real time? We can see their glucose levels, and we can measure their glucose levels during habitual physical activity using the Google Verily watch, during study exercises shown in blue, and we could quantify their heart rate and all sorts of other metrics. We know where they slept, what their body temperature was, and a whole bunch of other things. Now, remember, these people do a lot of activity, right? And I created this word bubble to try to represent the activities that were most common in the cohort. Walking was by far the most common activity in the cohort, as expected. 30% of all exercise events were walking. That's 4,813 walking events not yet analyzed. They did some biking. 8% of the data is from biking. And there's a fair amount of activities of daily living. 548 events of housework and cleaning, not yet analyzed. All we've analyzed so far are the study video exercise sessions, representing almost 3,000 exercise events. The first thing we wanted to see is whether aerobic, HIIT exercise, and resistance exercise have a different glycemic pattern in the wild. We knew from carefully controlled exercise studies in lab that resistance tended not to cause the glucose to drop, and aerobic did. And we did capture that in in the data. There was only a 9 milligram per deciliter average drop with resistance exercise and about a 20 mg drop with aerobic exercise. And these people are pretty educated on what to do with their diabetes when they do exercise. And we had enough power to determine difference between all these 3 exercise types. It's not that interesting, but aerobic does cause the glucose to drop more than interval versus resistance. What is really interesting is that it doesn't really matter what insulin infusion device you're on, you get the same effect. That startles me. That startles me. If you're on a $12,000 closed-loop system with algorithms designed for exercise, or whether you're on an insulin pen that is essentially free if you're prescribed insulin, the change was the same. And that is troubling. In all in all insulin delivery modalities, aerobic, you get more of a drop than interval than resistance. And look at the variance. We have tight error around the mean, but if you look at the 95% confidence interval on the patient variance, it's huge. Some patients, a lot of patients, have a rise during aerobic exercise, up to 30 mgs or more. Some have a massive drop, a drop more than 70 mgs in 30 minutes of exercise. The variance is a problem. But that's why we tackled the issue, is to get behind the variance in the first place. So looking at the 2,756 exercise video sessions, we now can explain much of the variance, which is exciting. And this is going to be shared at the ADA next week, and the journal, the paper is in draft form for submission. But I'll just hint to you that in addition to exercise type, we are now powered enough to say that these factors explain much of the variance in the glycemic response. And these factors are actually easy to quantify using either the person's own diabetes data or a few questionnaires before you customize an insulin pump designed for exercise. And there's a bunch of factors that did not become statistically significant. So I'm excited about these variables being integrated into new systems to tighten up that variance and to tighten up that hypoglycemia. And overall, our future goal in the next few years is to design activity wearables that can discreetly measure when the person's active and what activity they are doing, and integrate this information into an algorithm that'll then communicate to the insulin pump to fine-tune, individually customize that insulin delivery so that child or adult with type 1 does not develop hypoglycemia or hyperglycemia when they exercise. Because this data set is large and we can't mine it all ourselves, there's no way, it's going to be housed in a publicly available data set under what's called CETUS format. So you can download things like ambient temperature. Does that impact glycemia? Does step count impact glycemia? Et cetera, et cetera, et cetera. This is our gift to the research community. We can't do it on our own. I'd just like to acknowledge the funders for this project, the Helmsley Trust and the Jabe Center for helping to run the study, and for Dexcom and Verily Health for supplying some of the study product. And I could not do this study on my own. I need help from engineers. I need help from clinicians. I need help to solve this problem. And this is a diverse group of incredible experts across North America that are helping solve this problem. Thank you. We can take one or two questions before I introduce Dr. Nair, if anyone has any. Oh, gosh.
Only nice questions. Great job, Mike. Excellent
Thanks.
talk. I had two quick things. One is, we always think of insulin resistance as being a really bad thing, but as you so elegantly put it, there's so many variables in there. Do you think insulin resistance is maybe the body's mechanism to be homeostatic, to try to reduce those bouts of hypoglycemia?
Yeah. One interesting thing we just discovered related to that is that those patients with the highest A1C who tend to be more insulin resistant, they actually have less of a drop during exercise. So I think your hypothesis is probably true. Although we don't have gold standard measurements for measuring insulin sensitivity, unfortunately, it would be worth looking at.
Right. And your sleep data, so
yeah.
Poor sleep, poor or greater variability?
What was interesting, it's U-shaped. So the ideal sleep for this cohort seemed to be 7 to 8 hours a night. Less sleep, more hypo, more sleep, more hypo, which is fascinating.
Because I was just wondering, you know, if you have hypo and you've got the CGM, it's sending off alarms, you get a poor sleep, the next day you exercise, you go in, you know, and so does that end up-
Absolutely. I think that is a real problem that we see clinically, and we're seeing in the data set too.
So you'd be able to look over time at the same subjects were repeatedly getting spiraling downwards.
Yeah, we could. I guess we could. We could look at sleep metrics in an individual over the 4 weeks and see if things get worse or better based on their sleep patterns. We haven't done that, so that's a great idea.
Great.
Thank you.
Thanks.
Yes, sir.
Hi, Yun Tian from University of Virginia. So I wanted to ask you about that. So I read that last year, I'm sure you're aware of this, that Doug Melton's group had this human stem cell-derived islet therapy in 2 patients, and that was kind of wild. And I was just wondering whether, you know, you think that type of, you know, therapy instead of this, you know, traditional, like, you know, exogenous insulin giving therapy would kind of solve this.
I think the potential is there. They're using CRISPR genes to do implants now, and they're looking at the viability of the implants over the next year or so. And in fact, they're collecting data in Toronto at LMC, where I'm a research scientist. I think that has great potential, because if you just do pancreatic islet implants, you have to do rejection immunotherapy drugs.
Right.
And so it's hard to sustain that for your life. But if you implant it in this Gore-Tex sleeve where the immune system is basically ghosted to it, you might have a cure. I'm very hopeful. Very hopeful.
Thank you.
Thank you. All right. If we can pull up Dr. Nair's talk, I'll just do the intro. Dr. Srikumaran, often called Shri Nair, is a professor of medicine and consultant in the Division of Endocrinology and Diabetes Metabolism Nutrition in the Department of Internal Medicine at the Mayo Clinic in Rochester. He has his medical degree from State University in Albany and his PhD from the Council for National Academy Awards in London, very prestigious, MD, PhD. For more than 30 or 40 years almost, Shri has really led the field in the impact of insulin therapies on protein turnover. He really is a hero of mine and many in the field. I've seen him present before. He's got inspiring data. He also focuses on mitochondria and how insulin affects mitochondria turnover, and of course, the genetic risks for type 2 diabetes. Shri, you do all sorts of amazing things so well. It's an honor to have you here. Thank you.
Thank you, Mike, for that beautiful introduction. I truly appreciate that. I also want to thank David Hood for the invitation. Before I came to the hall, I told David that I made some change in my talk from my original one, because some people asked me, are you also going to talk about insulin? Then I said, I made the change because I thought I can convince all exercise physiologists insulin is probably one of the key factors involved in adaptation to the exercise program, three type of exercise program. Well, this was already shown by Michael, but this is really a favorite slide of mine because I started my research career. Robert Campbell, in the North Park Hospital, showed me this. I was impressed by the fact that even though diabetes is known as a disease of glucose metabolism, I think glucose is the first biomarker which was found. But looking at this, diabetes is not a disease of glucose metabolism alone. Look at such a, how do I go back? This one, really? No, I had to go back. Okay. So you can, the pre-diabetic period, the most pronounced thing was muscle wasting. And of course, there's not much fat also there. In fact, one of the old Greek physicians described diabetes as an affliction where you melt down your flesh in the urine. That's absolutely true. One of the, Michael mentioned about the studies I had done on protein turnover. One of the main findings we found was when you are insulin deficient, you really degrade muscle protein, and that comes as the amino acids and net nitrogen balance is affected by that. And so there is also something which I initially, when I found this, we did an entire calorimetry on these people. We found that their resting metabolic rate is high. They are consuming a lot of oxygen. That's a counterintuitive, because you may think that the oxygen is consumed for producing ATP, and that's unlikely to happen because no energy is stored in the body when they are diabetic. So that's another thing which I want to say that this muscle wasting occurs not only in type 1, it also occurs in type 2. They may be heavy, but they are also wasting their muscle. Well, many years ago, Craig Stump, who did a training with me, he later on became the chair of endocrinology at the University of Arizona. But unfortunately, 2 years ago, dear Craig died of a cancer. But this was one of the original findings of the Craig. What he did was he infused either insulin or saline into people and did muscle biopsies and also a lot of blood samples. What he found was that 4 hours, the saline group, of course, no change. The mRNA expression, this is using an old PCR approach. You can see the NADH4, cytochrome C oxidase 3, and COX4, all of them start going up, and pretty significant increase occurred by 8 hours, showing that insulin has an effect on transcriptional level on the skeletal muscle mitochondria. Then we use stabilizer tracers to really measure, I would say, translation rate, but protein synthesis rate, why this mRNA is translated into protein. When you do that, what we found was insulin fat increase mitochondrial protein synthesis. And of course, the insulin infusion usually reduces amino acid level because it inhibits protein degradation. You have to replace that amino acid. When that happens, protein synthesis also increased by insulin. So insulin is a key factor involved in mitochondrial biogenesis. And in the absence of insulin, this is an insulin deficient state. When you withdraw insulin from type 1 diabetes, what we did was we did the muscle biopsy, and then we, using a special column, we collected low molecular weight peptides, 28 kilodalton or lower, and which can occur only if in vivo protein degradation occurs. It's not a protein digestion. Also, non-tryptic peptides also looked at. They both give us an idea about in vivo protein degradation. What we found was there is substantial increase in vivo protein degradation. And when you look at their origin of those protein and functional pathways, we found that the main effect was on energy homeostasis and translational machinery of the tissue. Both are affected. So many of the mitochondrial proteins are degraded. Later on, I'm not going to show that study, we found in a mouse model that what happens is when you withdraw insulin in a mouse which was treated with insulin, diabetic mouse, it really increased the reactive oxygen species production. That damages the protein and degrades the protein. This predominantly happened to mitochondrial protein. If you do the insulin withdrawal, immediately after that, you don't see any degradation. But once this reactive oxygen species are acting on the protein, especially mitochondrial protein, there is increased degradation occurs. So I'm going to summarize a lot of studies we have done in this one. Withdraw the insulin, increase the oxygen consumption, and increase the fuel oxidation. But that fuel is not, oxidation doesn't result in ATP production. So uncoupling of mitochondrial oxidative phosphorylation occurs and reduces ATP production, but increases the ROS emission. More of the fuel oxidized, oxygen consumed is really diverted for reactive oxygen species production. And the result is it causes oxidative damage to proteins, and mostly mitochondrial protein. And oxidative damaged proteins are target for proteolysis by a ubiquitin ligase and autophagy pathway, which we have shown, and decreases mitochondrial content and mitochondrial function. I think Tom Hawk yesterday mentioned about a decrease in mitochondrial content in type 1 diabetes. This is likely cause. Even treated type 1 diabetes patient, as Michael Riddell has shown, they have periods of insulin deficiency, periods of excess of insulin, and glucagon is high, which again an energy-producing, energy-consuming hormone. So that way, they can get this continuous damage occur in the mitochondria, their mitochondrial content decline. This is also true in type 2 diabetes. So this is many of the studies summary I have shown here. So the question is how exercise counteracts adverse effects of insulin resistance on skeletal muscle mitochondria. I believe that this is a two-way traffic. The insulin effect on the exercise effect mitochondrial insulin sensitivity is the other way around also. Insulin action is necessary for mitochondrial biogenesis and also preserving the muscle mass and protein mass in the body. Well, this is a study done a few years ago. What we did was we brought in obese women, mainly women, these women with the diagnosis of polycystic ovarian syndrome. They are very insulin resistant. We matched them with lean controls, and what we found was when you do a muscle high resolution oximetry and measure this respiratory control ratio, which indicates coupling efficiency, and look at the ADPO ratio, which gives the phosphorylation efficiency, that's really showing that they have uncoupling occurring. And what we find is compare with the lean controls, obese women have significantly lower coupling efficiency and phosphorylation efficiency. And you can see that when you put them, we randomize these obese women to 3 months of exercise program or sedentary period. What you are seeing is you can see that this respiratory control ratio significantly increased in the exercise group, did not change in the sedentary group. Same by ADP ratio also increased. So exercise can reverse this thing in insulin-resistant people. And further, this is showing that we also look at the reactive oxygen species production, which is mostly by H2O2 emission when the muscle mitochondrial preparation. And you can see that it was significantly higher in the obese people. And when they are exercise compared to their sedentary controls, they have significantly significantly they reduce the reactive oxygen species production. So oxidative stress is significantly reduced by aerobic exercise program training for 3 months. And this is showing the, in fact, catalase activity increase also. And we looked at DNA damage, DNA oxidation by 8-oxo-DG by a mass spectrometry. What it shows is that you can see even 3 months of exercise has significant reduction of oxidative damage to DNA. So oxidative stress can be reversed by aerobic exercise program. How exercise counteract the adverse effect of insulin resistance on brain is not only muscle. Yesterday, Bruce Spiegelman mentioned about increasing prevalence of dementia and Alzheimer's disease in people with diabetes, type 2 diabetes, and also insulin-resistant people. This is becoming an increasingly a problem as people are aging longer, and that in combination with insulin resistance. We are doing a human study using a phosphorus MR spectroscopy and functional MRI and all. But meanwhile, the tissue we can take from mouse. So what we did was we use mouse and induce insulin resistant by high-fat diet. And then controlled mice, we also exercise them, and also high-fat diet mouse also we exercise. This is showing the mitochondrial ATP production in the cortex. That's where you get maximum tissue. We had a hippocampus. There we see the mitochondrial enzyme we measured. What we are seeing is mitochondrial ATP production rate significantly reduced in mouse who are on high-fat diet and insulin resistant.
But this can be completely reversed by aerobic exercise of 6 weeks. This is running on a wheel. And of course, aerobic exercise also increase in other, the chow-fed mouse also. So aerobic exercise can increase mitochondrial ATP production in the brain. Previously, we thought that insulin doesn't have much of an effect in the brain because the glucose metabolism in brain doesn't require insulin. But insulin receptors are rich in certain areas of the brain. So happen, these areas of the brain exactly the areas where Alzheimer's disease is affected. So because we know that in muscle, insulin can affect mitochondrial biogenesis, you want to look at the brain. That's why we specifically look at the areas where insulin receptors are high. I'm not going to show the data, but midbrain, insulin receptors are low. And high-fat diet did not have any effect on mitochondrial function there. It's really the areas of insulin receptors rich, it is happening. And here, of course, what we are showing is a reactive oxygen species production, H2O2 emission. You can see significantly go up on high-fat diet, and exercise completely prevented that happening. So yes, exercise can benefit brain also. Probably I'll also show another slide showing that this is hippocampus in the brain, and you can see that we look at the proteomic analysis. And here what I'm showing is this is an increase in the protein oxidation occurring in the hippocampus region, high-fat diet mouse. All the red are mitochondrial protein. So mitochondrial proteins especially damage the high-fat diet in the brain. And this is if you exercise them. It is all corrected, or it can be prevented happening. So exercise can prevent various changes occurring in the brain in terms of protein. And this, again, these are happening in the hippocampus, one of the main areas involved in the memory and Alzheimer's disease. Well, we know that aerobic exercise, mitochondria is so important, but resistance exercise, sarcomeres, the contractile apparatus is involved. And I just wanted to show some studies done many years ago. This is showing that if you really look at the young and middle-aged and old people, their ability to synthesize muscle protein, mitochondrial protein, decline by almost middle age, and in concurrent to decrease in the oxidative enzymes. So almost by 45 to 50 years, your ability to synthesize mitochondrial protein substantial decline, especially if you are sedentary. And again, we showed that in another study, in fact, mitochondrial DNA abundance decline, proportional to the mitochondrial capacity to produce ATP also decline. And we also found a reasonable correlation to age with the oxidative damage to DNA also occurring. So a key factor in terms of DNA damage also is related to mitochondrial dysfunction. Can exercise training counteract age-related decline in muscle mitochondria? Well, this is a study, Ian Lanza, who did a postdoctoral fellowship with me, but he's a colleague, independent investigator in Mayo Clinic. What Ian did was he recruited people who are highly trained. That means at least 4 years they are doing long-distance biking, and at least 4 to 5 days a week. Young and old people the same way. Again, young and old sedentary people who are healthy otherwise. There's no other predisposing illness. So Ian looked at their mitochondrial DNA abundance. These young people, those who are aerobically exercising, there is an increase. But, but on the other hand, if you look at sedentary old people, they have, as expected, significantly lower mitochondrial DNA. They increase their mitochondrial DNA reduction can be prevented by this long-distance exercise program going on for more than 4 years. We don't have any data on the short-term exercise. But if you look at this one, this is almost the same as sedentary young people. And if you look at the ATP production rate, you can see that young people, there's an increase occurred, and there's a reduction occurred in old people who are sedentary. The increase occurred. The mitochondrial muscle ATP production is very similar to young sedentary people. That's not a bad deal if you are 70 years old. And if you get the same mitochondrial ATP production as a young sedentary person, that's a good deal. You can achieve that by maintaining exercise program. Well, simply want to show that it's also change in the PGC alpha, NRF1 and the TFAM, they all changed. Well, we also, this study and many other studies, we did a proteome analysis in the muscle. Here, specifically mitochondrial protein, this is ex vivo labeling and then mass spectrometry and then we looked at mitochondrial protein. What you are seeing here is, this is old sedentary comparison with the old trained people. You can see old sedentary people have all this, blue is the glycolytic, other one is oxidative. They are, all these ones are significantly lower. One of them is citrate synthase there. And this one is old trained and old sedentary. We can see almost the same thing. Old trained people have significantly higher all of this protein. This not only age alone matters. And here is young trained and young sedentary. Yes, young trained people also have increased oxidative proteins there. And this is old trained comparison with, comparison with young trained. The difference is very little. Most of them is corrected. So basically showing the protein is a functional molecule and the changes in the protein which occurs with aging can be prevented if you really engage in exercise program. Well, also want to see insulin resistance, which is, we thought insulin resistance occurs with age. This and many other studies we have shown that that's not entirely true. Just want to show that this is hyperinsulinic euglycemic lamp. The glucose infusion rate will tell you whether your body is more sensitive to insulin, especially muscle. What it shows is when you need more glucose, you are more insulin sensitive. These two groups are young trained and old trained. These two are young sedentary and old sedentary. So trained, old and younger people have a similar insulin sensitivity, peripherally and in liver. I'm not going through this one, but hepatic glucose production also we measure. Insulin resistance of age is related to sedentary lifestyle. So you can prevent diabetes. Okay, that was more of a cross-sectional study. We did another study where we brought in young and old people, both men and women, equal numbers, and who are perfectly normal. And their lean body mass, they are not BMI, everything, they are pretty matched. What we are shown is the effect of exercise program. We did the three type of exercise. It's really high intensity interval training for 3 months, resistance training, fairly high intensity, and combined training is what is recommended by American College of Sports Medicine, more moderate level of aerobic and resistance training. One thing which is evident is that it doesn't matter what exercise you do, there is improvement of insulin sensitivity based on glucose disposal. You can see the high intensity and resistance both improve almost the same level. And in young people, combined training also improve the same level. But older people, as many people have shown, the intensity of exercise is important to reverse many of the changes occur. They didn't find, they had shown a 10, but they didn't show significant effect. And if you look at VO2 max, of course, VO2 max increase in the high intensity interval training, maximum increase in the young people. Old people lag behind. Resistance training did not significantly increase, combined training increase. And interesting enough, if you look at the mitochondrial respiration, it increased equally young and old. In fact, if you look at percentage increase, to start with the young were lower, old were lower, they have higher increase occurred. But even though we have shown many studies that there is a clear correlation with the maximum ATP production capacity and VO2 max, but here is an exception. I believe that's a cardiac factor. The cardiac output may not be catching up in the older people, and also perfusion may be another factor. But then if you look at their muscle strength, as you would expect, the effect was more on resistance exercise. Even though lean mass increase in the high-intensity interval training, they did not significantly increase muscle strength. So you need resistance training to improve the muscle strength. That's what this study has shown. Even though fat-free mass can be improved by high-intensity interval training also. So aerobic and resistance exercise enhances insulin sensitivity, both of them. And the aerobic and exercise is critical for mitochondrial and VO2 max, and resistance exercise critical to maintain muscle mass and strength. Well, we looked at many analyses done in this study. Identification of universal exercise response genes. We found 55 unique genes with exercise, regardless of the age and type of exercise. And we did some, what we call the upstream regulator analysis. What we found is most remarkable effect is on angiogenesis. So exercise, one way it improves function, mitochondrial function, I believe, is improving the perfusion to mitochondria. So you can take oxygen and substrate to the mitochondria. Mitochondria can function better. And also, insulin delivery to tissue also is important, which again evidence that perfusion of the tissue helps. So that seems to be an important thing happening with exercise. And this one I just want to show mainly this one. The thing is that oxidative damage and also deamidation, which is one of the pre-apoptotic state, many proteins are in the deamidated state. Sedentary group, there is increase occurred, but both exercise program prevented that happening. So oxidative damage can be prevented. And another important thing is that if you look at the proteins which are increased, some of the key proteins increase are in the ribosomal protein. So that's the reason, even though 3 months exercise, we found that many of the increase in the transcriptome were much smaller than increase in the proteome. That means we also found the protein synthesis increase. That's because enhancing the machinery involved in the protein translation, the RNA translation of the protein. One of the other things, see, this is a study from different study. What I want to show is that if you look at ATOX or DG, sedentary people and trained people look at, there's a significant difference. So if you are aerobically trained, there's less DNA damage also occurs. So resistance exercise and skeletal muscle contractile apparatus, they are the important, so we looked more into that in the resistance exercise group. Many years ago, I think we did this study where we looked at myosin heavy chain synthesis rate and also mixed muscle protein synthesis rate using a tracer approach. What you are seeing is 3 months of resistance exercise in people 55 or above. You can see that myosin heavy chain synthesis rate goes up, and the mixed muscle protein synthesis also goes up. Control group change occurs. So resistance exercise increases hypertrophy partly by increasing muscle contractile protein, by increasing their synthesis rate. And but another important question is what happens in the energy metabolism? We all know that mitochondria is involved in that, but mitochondria, to get ATP from mitochondria takes some time. But quick action of the contractile muscle action require immediate supply of ATP. And that can be provided by, you can see the one glycolytic pathway, you can get 4 ATP, and ATP can be immediately produced. Increasing evidence by many laboratories showing that glycolysis is a possible way resistance training exercise is happening. And this one, we are very partly involved in this study with George Rua, who is here, an elegant study where George Rua and Bruce Spiegelman. And there, what has been shown is one of the isoform PGC1 alpha, is PGC1 alpha 4, is involved in the hypertrophy. It is a hypertrophy gene. So the question we asked was, PGC1 alpha 4 regulates muscle hypertrophy induced by resistance exercise. Does it also increase the glycolysis induced by resistance exercise? It makes a more teleological sense. The same gene does both of them. So what we have shown is, here you can see that the muscle enzyme activity, the glycolytic enzyme hexokinase, and phosphofructokinase, which is one of the rate-limiting enzyme, LDH, one immediately after exercise, this non-exercise leg and exercise leg increase of this enzyme occur. Even after, for 72 hours after the 12 weeks of exercise, hexokinase increase, LDH also increase, supporting that these activities are, enzyme activities are increased by resistance exercise. This is simply looking at the gene changes occurring, glycolytic genes changes to resistance exercise. And we wanted to look at the mRNA expression. 3 months exercise, 72 hours after the last bout, mRNA levels are not significant. But the protein levels, as Bruce Spiegelman mentioned yesterday, is not easy to quantify. But we see a definite increase occurring 3 months after exercise, PGC1 alpha 4, very little for PGC1 alpha 1. So what Jin-Ho Ko did, this is a study by Jin-Ho Ko and Mark Pataki, and what he did was he really overexpressed PGC1 alpha 4 in myotubes, and here using the seahorse approach, what is shown is summary of this one is increasing the glycolysis and glycolytic capacity, significant increase of PGC1 alpha 4 expression in the myotubes. And you can see that if you myotubes, these glycolytic proteins are increased by PGC1 alpha 4, not that level at all by the PGC1 alpha 1. So this is oxygen consumption. You can see that this PGC1 alpha 4 doesn't increase oxygen consumption. So it's showing that PGC1 alpha 4 increase the glycolysis, it's anaerobic glycolysis, not oxygen dependent. So this showing that hexokinase activity also increase. So exercise training, in fact, insulin sensitivity, PGC1 alpha 1, it cooperating with PPR gamma, it increases oxidative metabolism, it reduces reactiosin species production, which also may contribute to improvement of insulin sensitivity. And PGC1 alpha 4 increase hypertrophy, but it also increases glycolysis metabolism also. So I'm going to stop it here saying that as we are all getting old, the only way to prevent getting dementia is exercise. This is actually taken from American College of Neurology with their recommendation. Improve memory for people with a mild impairment. Only solution for no drugs so far is aerobic exercise. I also want to thank people who really did the work. Many of the people, Mark Pataki was trained with Greg Cartee, and Jin-Ho Ko, who is now a faculty back in South Korea, and many people in this group, and of course with the NIH grant and the Mayo support. Thank you so much.
It's a little humbling to see the cumulative impact factor of what you presented, maybe like 590, where what I presented was an impact factor of 0 so far, but very impressive collection of data. We've got time for a couple questions before I introduce the next speaker.
Yeah, fascinating talk. Thank you very much, Henning Wackerhage. So in your last study, you showed essentially that sort of the hypertrophying muscle fiber switched on glycolysis. And this is similar what happens in cancer. This is a Warburg effect in cancer. So because Otto Warburg found that sort of when cancer cells have switched to glycolysis, and that is essential for growth. So do you think something similar is going on in hypertrophying muscle?
Sorry, can you ask that question again?
Yeah, Otto Warburg.
Oh yeah, you know.
Yeah, so the Warburg effect. Do you think?
Cancer, of course, glycolysis is the main energy provider. But now increasing evidence showing that, in fact, the mitochondria is producing a lot of energy, and that's consuming a lot of glutamate pathway, alpha-ketoglutarate to get into the cycle and providing energy also. Yes, it may be needed for rapid growth. And
okay, thank you.
That's probably likely. And also, we don't know the rapid growth in many stages it's needed. So really a combination of these two. Okay. Yes.
The data you provide with the oxidation of proteins with a high-fat diet and the impact of exercise is phenomenal. my question is that, do you know where are those proteins in? Are in neuronal cells or astrocytes, microglial cells? And then if indeed they are reduced in the neuronal cells or other cells, what is the possible mechanism? Is this due to activity or due to some other humoral factors in that cause the changes?
Well, that's a very important question. Working in the area of muscle for many years, I realized that it's a very unique organ, brain. It has so many defense systems and multiple different cells, and it's not very simplistic approach. You're absolutely right. What we have done is really the whole hippocampus or cortex. But we have done some neuronal cell lines. There also it's happening. And, and also I must say that another interesting thing is that if you make them insulin deprived, they are not as much affected by insulin resistance. The reason is insulin deprivation increases the lactate production and also ketone production. And the transport system in the blood-brain barrier is upregulated, so that you can transport a lot of lactate and ketone to the brain, and brain can use them. That also has some antioxidant effect, increase the antioxidant defense system in the brain. So it's not like muscle. Brain protects itself by different ways. So it's both, again, if you ask me what cells, I don't know. We are getting into that. That's probably next 10 years' work.
Thank you.
Thank you so much, Dr. Nair.
Thanks very much.
I could abbreviate the next speaker's brief biography into one word if I wanted to. The word would be prodigy. You really are a prodigy. Having been mentored by Eric Richter, Bruce Spiegelman, and Paul Gregovic in mounting such an incredible career over just 14 years is inspiring. Liki is currently associate professor and head of the molecular medicine in cancer and aging group at the University of Copenhagen. She's worked in basic science for most of her academic career. She's trained with Eric and Bruce and published with Amira, and Paul has been a mentor. Just an incredible pedigree, unbelievable. We are so pleased to have you here. Liki, take the stage.
For that very kind introduction. I think I have indeed been very fortunate to have some of the best supervisors in the world and mentees, so I'm very grateful for that. And I'm also very grateful to the organizers for the opportunity to share some of the work that we do here today. I work in the molecular metabolism in cancer and aging group, where we try to understand at the molecular level how skeletal muscle mass and metabolism are regulated in health and disease. But before I dive into that, I want to acknowledge the group that I work in that makes every day wonderful to come to work, as well as our fantastic collaborators and funding, and without whom none of this would happen. And I would like to highlight that Stefan and Fung are here, so I hope that you will find them and say hi to them. They're very nice and very smart. Okay, exercise is likely the most effective health span promoting intervention in humans. And that has been documented in numerous epidemiological studies and randomized controlled trials that has really shown us how beneficial exercise is in preventing multiple diseases. And with skeletal muscle being central to metabolic control and mobility, and us being very muscle-centric in our behavior and our capabilities, we have studied this with a focus on skeletal muscle. So today I will speak about the acute effects of exercise with a focus on how exercise increases glucose uptake and the contributions to that from the Rho GTPases. And I will also speak about some chronic exercise training effects whereby Rho GTPases might be implicated in non-canonical muscle mass regulation. And in the end, I will touch upon how mitochondrial mRNA processing might also be important for exercise adaptations. Yep. So we have these acute exercise effects. And so overall, I think the beneficial effects of exercise can be grouped into two. And that's one of the acute effects, and then the chronic exercise effects. And part of these acute effects is what is happening exactly when we do the exercise. So if I go for a run, I will have a marked increase in the glucose uptake by the muscle, and there will also be other different muscle adaptations that occur instantly. And that will happen to all of us regardless of our exercise training status. Another thing that happens immediately after that one exercise bout is that there is an upregulation of insulin sensitivity in the muscles that have been doing the work. So these are very acute and very beneficial adaptations to exercise that occur regardless of the exercise trained state. And then we have all of the different adaptations that occur slowly in response to multiple exercise training bouts that here is illustrated in this yellow curve. That include upregulation of mitochondrial proteins and increased mitochondrial function that I'll speak a bit about, and then also an increase in muscle mass and strength. I also want to mention that the beneficial effects of exercise, of being in an exercise trained state, is probably also due to the fact that if you're an exercise trained individual, you'll be spending a substantial amount of your time in these acute beneficial phases. Yeah. So now I'll speak about this acute phase where glucose uptake is happening in the muscle. And we know that during exercise, carbohydrates are utilized by the working muscles, as we heard beautiful yesterday in Mark's award lecture. And a fundamental response to exercise is then the transport of glucose into the skeletal muscle. And the mechanisms underlying this was really made possible to study by the cloning of the GLUT4 transporter in the 80s. And now we know that the insertion of this GLUT4 glucose transporter into the plasma membrane is necessary for the glucose to cross the lipid bilayer of the membrane and enter the cell. And this GLUT4 translocation is actually a necessary step both in insulin but also in exercise-induced glucose uptake. And further studies going into the 90s, here by Laurie Goodyear's group, has shown that the mechanisms by which insulin and exercise gets GLUT4 to the plasma membrane are distinct. So here it's shown that in the Mirco mice, which is the insulin receptor knockout mice, there is a reduction in insulin-stimulated glucose uptake because the insulin cannot signal to GLUT4 translocation, whereas this does not occur with exercise-induced glucose uptake. So that really shows that these signaling pathways are distinct. Another thing that has told us that is also the study showing that if you add exercise or muscle contraction and then insulin on top, these two effects are additive, so you get an additive response on glucose uptake. That is good news if you are a type 2 diabetic. It is bad news if you're a type 1 diabetic, because then it can be difficult to manage. But if you have insulin resistance, here shown here in these type 2 diabetes, and these are, I don't want to call them normal, but that's what they did, I guess, back in the days, but healthy controls. The induction by exercise of GLUT4 through the plasma membrane is similar in healthy controls and type 2 diabetic subjects. And that is important because it can help people with type 2 diabetes and other insulin resistance condition managing and getting glucose away from the blood and into the myocytes. Okay, so exercise seems to be able to bypass insulin resistance. But I want to really briefly touch upon the insulin signaling cascade, because like exercise, insulin results in the translocation of the GLUT4 vesicles to the plasma membrane. And it does so by this really well-studied, very complex and highly regulated intracellular signaling cascade that has traditionally been defined via this AKT axis. And a lesser described part of this signaling pathway is a pathway where RAC1 induces reorganization of the actin cytoskeleton, which is necessary for the GLUT4 transporter to be inserted into the plasma membrane. And this work on RAC1 and the actin cytoskeleton has really been pioneered by Amira Klipp's team here in Toronto. And then we have followed it up afterwards. And we have really shown that this component is as important as the AKT component to regulate insulin-stimulated glucose uptake. And what we have also found is that actually, in contrast to the AKT signaling pathway, the RAC1 signaling pathways seem to be also activated by exercise and seem to be also necessary for exercise to fully induce glucose uptake. And I'll show you that now. But first, I just want to introduce you to the Rho GGPases a little bit. And the ones that we have present mainly in skeletal muscle is RAC1, CDC42, and Rho A. And we have mainly studied RAC1. And Rho GGPases are really important for so many different cellular processes. And because of that, and I might be a little biased, but I think it's incredible that we have not studied them more in skeletal muscle. But we're trying to do this now, and I'll show you some of our data. So in collaboration with Thomas Jensen's group, we have looked at RAC1 activation in response to exercise. And RAC1 cycles between a GDP-bound inactive state and a GTP-bound active state. And in response to uphill walking in the skeletal muscle, there is an upregulation of RAC1 GTP binding in the soleus muscle and here in the gastrocnemius muscle. And when RAC1 is active, it can bind to its downstream target, which is PAC1 or PAC2 in skeletal muscle. And that elicits a conformational change of PAC, which results in an autophosphorylation of this kinase. And that is a phosphorylation that we have used as a surrogate measure of RAC1 activation because it is quite difficult to measure RAC1 GTT binding due to very fast hydrolysis. And we have seen that also PAC phosphorylation increases after this exercise bout, suggesting that RAC1 is active. So I'm from Copenhagen, and we love to bike. It's usually very pleasant, sometimes not. But we bike all of the time. So of course we had to check if bicycle exercise would also activate RAC1. So we had 4 of the students at the time in the lab cycle on a bicycle, and this is a project that has been driven by Stefan Ravn. And then we had Professor Erik Richter coming, taking a muscle biopsy before and after this bicycle exercise bout. And then we studied PAC phosphorylation, and I think you can see that in most of these students, there was an upregulation of PAC phosphorylation. And Stefan has told me that this is him, so he's a very nice responder. Yeah, so that's good. So really, RAC1 is activated by exercise and also by many different exercise modalities in humans. And luckily, that's also the case in mice. And the reason that we like that is because then we can start to study some of the mechanistic underpinnings and roles for RAC1 in exercise-induced glucose uptake. So we generated these inducible muscle-specific RAC1 knockout mice, and then we ran them on a treadmill for 20 minutes. And before we started the exercise bout, we injected isotopic tracers so that we could trace where the glucose would be going in the body during this exercise bout. And what we found was that in glutamate-controlled mice, there was a nice and robust upregulation of glucose uptake in response to the exercise, but this was partially inhibited in the RAC1 knockout mice. So that suggested to us that RAC1 is necessary in order for exercise to fully stimulate exercise-induced glucose uptake. So what could be the activating mechanism? And this took a long time to figure out, but I think maybe we know it now. So we know that muscle excitation results in release of calcium from the sarcoplasmatic reticulum, which results in cross-bridge formation and turnover of energy that is sensed by AMPK. So we have looked at whether we thought AMPK was activating RAC1, but it does not look like that because ACAR or other compounds that activates AMPK does not seem to activate RAC1. But there is another component that I think is a little too overlooked to contraction, and that is the mechanical stress that is put on the plasma membrane during cross-bridge formation. And that is sensed by the integrins or the DGC complex, a large complex here at the plasma membrane, and that can activate RAC1. And we have shown this numerous times now, but I think the most or the best evidence of this is just this figure here, that if we take, here it's EDL muscles, and we stretch them in incubation chambers, and we stretch them to the same tension that they will reach if they are electrically stimulated to contract, then we see this upregulation of PAC phosphorylation, which is a surrogate measure of RAC1 activity. That is, of course, not present in the RAC1 knockout mice. So we think that RAC1 is activated via the stress that is put on the membrane during exercise or contraction. And we have then tried to understand a little more into detail what mechanisms could it be that allows for this RAC1 activation. And this is work that was driven by Lisbeth. She's a postdoc on the team. And it's work that started when I was in Bruce Spiegelman's lab, which is very, very long time ago. So I'm a little embarrassed we haven't published it yet, but hopefully soon. But what we found was that, so we checked the RAC1 interactome. And we found that especially when RAC1 was in its inactive TDP-bound form, it was heavily bound to this ARC-DIA protein, which is called GDI-alpha. And we were very interested in this because in non-muscle cells, this has been shown to be a potent inhibitor of RAC1. So we moved on with this and with the hypothesis that, that GDI would inhibit RAC1, so that if we could remove GDI from RAC1, RAC1 would be active. We also have some proteomics and phosphoproteomics data to show that GDI can be activated during exercise, or can be phosphorylated during exercise, so maybe this could be a mechanism of dissociation, but that's something we still need to understand better. So what Lisbeth did was she knocked down GDI-alpha here in L6 myotubes, and according to what we thought would happen was that we saw that there was a marked increase in RAC1 GTP loading when GDI was not there to inhibit RAC1. So this could be a potential mechanism by which RAC1 could be activated, that there is an increased dissociation from GDI in response to exercise. Yeah, so we have also wondered about the interaction between RAC1 and AMPK and the relative contribution to glucose uptake. So in order to try and understand this better, we generated double knockout mice, so mice that lacked or had an overexpression of a kinase mutant of AMPK, and to cross them with inducible RAC1 muscle-specific mice. And these are the 4 groups we have. So, and then we incubated soleus and EDL skeletal muscle in incubation chambers, and then we could electrically stimulate them to contract and use isotopic tracers to trace the glucose uptake of these muscles. And in control muscles, there was a nice induction in glucose uptake in response to the contraction that was partially inhibited in the RAC1 knockout mice and the AMPK-KD mice. And what we thought was interesting was that there was an additive effect of adding RAC1 and AMPK inhibition to the system. So we think that RAC1 and AMPK do work in parallel on glucose uptake. But I also want to say that there is still an increase in glucose uptake even in the complete absence of these 2 pathways. And what that is, I'm not sure, but hopefully we'll find out. And this has also recently been reproduced again here, instead of having the AMPK-KD, now with the TPC1D4 and D1 knockout combined with RAC1 inhibition by Al-Hassani's group. So we don't think that they are up or downstream of each other, but work from Carlos in Copenhagen as well, and Thomas Jensen's group has suggested that maybe RAC1 acts via ROS production, because RAC is a really important part of this NOX2 complex that is necessary for ROS production in cardiomyocytes and also in skeletal muscle. So we have, with Stefan, Ravn again, and Carlos, we have used a recombinant adeno-associated virus to overexpress a constitutively active form of RAC1 in the one muscle, and then injected with a control virus in the contralateral leg to study exercise-induced ROS production. And so the staining that has been done is DCF fluorescence, so it's a redox-sensitive dye that measures total oxidants. And with exercise, there is an upregulation of total oxidant production, as has been shown before. But if we have an overexpression of this constitutively active RAC1, this is markedly increased. So we do think that RAC1 hyperactivation really promotes the production of reactive oxygen species, and Carlos has really nice data showing as well, and he will show you them tomorrow, that in the RAC1 knockout mice, ROS production does not occur in response to exercise. So ROS production does seem to be important for glucose uptake, and this is again evidence from Carlos showing here in a mouse with a point mutation of the P47 subunit here of the NOX2 complex, rendering the NOX2 complex non-functional. And like in the RAC1 knockout mice, that markedly reduces exercise-induced glucose uptake. So we think that RAC1 mediates glucose uptake via the production of reactive oxygen species. Good. So just a summary about the acute exercise effects. We have shown that multiple exercise and contraction modalities will activate RAC1 in skeletal muscle. And exercise activates RAC1 in parallel to MPK. That's what we think so far. Likely via stretch-mediated activation. And our data point towards that RAC1 is required for exercise to fully stimulate glucose uptake, and that's likely via the production of reactive oxygen species. Okay, so another acute effect of exercise that has been very nicely described and shown by Professor Richter and others, and I'm sure we'll hear more about that from Greg Cartee's talk soon, is this increase in insulin sensitivity. And because we'll have a talk about it, and we have not done so much work on it, I'll just very briefly go over and go away to look more into some of these chronic exercise training adaptations. And here we'll focus on muscle mass regulation. I'm sorry. So what is really regulating these chronic exercise adaptations is probably a marked upregulation or downregulation of proteins implicated or transcription here. And transcription is very heavily regulated by phosphorylation events. So in this very nice study that has been led by Professor Richter and Keynes, as well as Benjamin Parker, and he has a poster on this, they have looked at the phosphoproteome in response to different exercise modalities: endurance exercise, sprint, and resistance, in just one single exercise bout. And they've looked acutely after, but they've also looked into the period of recovery, which has been interesting to us. And what I just want to show here, and I really recommend you go to Ben's poster for more information on this, is that some of the most enriched pathways in the recovery from sprint as well as resistance exercise training or exercise bout was signaling by Rho GTPases. And this is something we have looked a little further into, and we're now trying to understand the implications. And we think that RAC1 could be implicated in muscle mass regulation. So in the heart, angiotensin II results in hypertrophy because it activates ROS production via RAC1 that then activates this downstream MAP kinase signaling pathway to induce cardiac hypertrophy. And in angiotensin-treated RAC1 cardiac-specific knockout mice, this induction of hypertrophy does not occur. So we have been interested in trying to look at, could it be that also exercise training induced hypertrophy would not occur in our muscle-specific RAC1 knockout mice? So Stefan has placed some RAC1 knockout mice on a treadmill and let them run for 12 weeks. And in our hands, that does increase gastrocnemius muscle mass as well as tibialis anterior muscle mass. And this effect does not seem to occur in the RAC1 knockout mice. And I just want to say that they run the same distance, and also the exercise capacity improvements with exercise seems to be fairly similar as well. But it does look like RAC1 could be implicated in muscle mass regulation. And Stefan has also looked a little more into what could the mechanism for this be. So he stimulated RAC1 knockout mice in one leg with electrical contraction, leaving the other leg as the control, and then studied downstream signaling in recovery after 4 hours. Stefan looked at mTOR signaling, and that was induced in recovery, but no changes with the RAC1 knockout mice. However, when looking downstream here of P38 MAP kinase, here he checked protein 27 phosphorylation as well as CREP phosphorylation. The induction with a prior contraction seemed to be reduced in the RAC1 knockout mice. So this could be that RAC1 is mediating hypertrophy response in an mTOR-independent manner via this pathway here. And if we're looking back on the cardiac hypertrophy model, that could be mediated via reactive oxygen species production. But this is something we need to, I think, look further into. Another piece of evidence that suggests that the Rho GGBases, or RAC1, could be implicated in muscle mass regulation is data from Lisbeth Møller. And she has used recombinant adeno-associated virus to downregulate GDI-alpha in skeletal muscle. So remember that if we downregulate GDI, we get an upregulation of RAC1 activation. And she's done that in one leg, leaving the other contralateral leg as the control. And just by knocking down GDI-alpha to this degree here, she did see an upregulation of muscle mass in these muscles, indicating that there is some regulation on this pathway. Good. So now I'm going from Rho GGBases and into something very different, because another thing that has come up in recovery is a marked regulation or marked enrichment in the phosphoproteome of regulation of mRNA processing. And this has been an interest, a newer interest on the team, and it's work that has been driven by Fung Pham, who is also here, and she has a really exciting poster on this. And she has been interested in this protein that sits inside the mitochondria called SLRP. And SLRP binds to the mitochondrial RNA. So our mitochondria are really a unique organelle in that it contains its own DNA that encodes for only 13 proteins. And in order for that DNA to be transcribed, the mitochondrial RNA needs to be stabilized and be transcribed here in the mitoribosomes for translation. And we have generated, or we have shown that this SLRP protein is one of the most upregulated proteins in our proteomics, and also here by Western blot with exercise training, so chronic exercise training. So we've generated, together with Niels Göran Larsson's group at the Karolinska, these SLRP knockout mice. And what is really interesting, you can see it here in the red, is that in nearly all of these mitochondrial DNA-encoded transcript, there is a marked downregulation of them in these SLRP knockout mice. And that has quite a striking phenotype, but I'm just going to show you one of them here, which is, so this is a confocal microscopy image of a single fiber that is where we've stained the mitochondrial network structure using a membrane potential probe, together with Carlos again. And I think you can easily appreciate that these mitochondria are highly fragmented. And we're now trying to understand what could be the underlying mechanisms for this. Okay, so in summary of this chronic exercise training part, we think, or we know that Rho GGBA signaling is enriched in recovery from exercise. And we believe now, based on these data I showed you, that perhaps RAC1 plays a role in muscle mass regulation, independent of mTOR. And we've also have some new data showing that mitochondrial mRNA processing by SLRP could regulate mitochondrial function in skeletal muscle. So I think it's really incredible how far we have come in understanding these complexities of exercise adaptations. Yet I think we're only at the tip of the iceberg, and many exciting discoveries in exercise biochemistry lies ahead. So thank you very much for your attention.
Leaky, we are lucky to have you at the tip of the iceberg looking at this for us. Fantastic presentation. We've got time for a couple questions. I can ask one while we wait for people to head to the mic. You showed convincingly that RAC1 plays a pretty critical role in the normal physiology adaptation to exercise and training, acute bouts. But where's the evidence that it has a role in the pathophysiology, let's say, of a chronic disease like type 2 diabetes? So it has like a therapeutic potential.
Yeah. I mean, the evidence we have is that insulin-stimulated RAC1 activation is markedly reduced in type 2 diabetics, also in obese subjects. Whether it has therapeutic potential, I'm not sure of, not until we figure out a way to target skeletal muscle with a drug, because it's also an oncogene, so it would, yeah.
And it's I guess, H2O2 production, is that concerning therapeutically or not, do you think?
Probably. Yeah,
yes, sir.
Great talk. Just have a quick question. So you said like RAC1 might be involved in the muscle mass regulation. So I know you published some papers using cancer-mediated cachexia. So have you looked at this RAC1 activity in cancer cachexia? Do you have a plan for that?
Yeah, we have not looked yet, but we think it's really interesting. So far we have looked at insulin sensitivity in cells that have been treated with media from breast cancer cells. But that's not a cachexia-inducing phenotype. But they also become insulin resistant. And here we see a marked reduction in insulin-stimulated RAC1 activation. But we have not looked yet. But yeah, it's definitely something we would like to do. Yeah.
Prof. Fabreo?
This is probably a rather naive question from someone who doesn't know much about GLUT4, but I'm going to ask it. So, has anyone ever looked at whether or not the vesicle from a patient with type 2 diabetes versus a control, or from a muscle that's been insulin stimulated or contracted, is the same? So is the component of the vesicle to get GLUT4 to the membrane, either in those two comparisons, does anyone know if there are any differences in the vesicle? So I've been listening to you guys talk about the differences all the time. As far as I can understand, you still don't know why insulin and contraction are different. Is that right?
Yes, I think that's completely right. I think also, I can't answer your question. But if you're also alluding to this increase in insulin sensitivity after exercise, that's not unique to exercise. It's also if you, for example, stimulate with other stimuli. For example, if you stimulate with insulin, this is something Holossi has found, and then afterwards stimulate with insulin again, there is also an insulin sensitizing effect to the second insulin stimulation. But I don't understand the mechanisms, and
okay,
maybe someone else do. But,
so I'll ask a follow-up question. Is there any evidence that you can have GLUT4 that's not vesicularly bound?
Not to my knowledge.
The prodigy has a mentor here about to take the mic. Dr. Clip.
I just think it's a really good opportunity to say the little that is known about the compartments that respond to insulin or to exercise. And the very first papers, when we and then the Goodyear group isolated a fraction enriched in an insulin-responsive component, at least in our hands, that compartment was not responsive to exercise. When we did the exercise, there was a gain in the plasma membrane, but we couldn't identify a distinct compartment that was responding. Now, fast forward about 25 or 30 years, and from kinetic measurements, partly done by Holman, partly done by us, in systems in which you emulate the exercise pathway by manipulating the signals, so activating K, et cetera. I think the evidence is pointing to insulin promoting an exocytic movement of a particular compartment, that is the one we're chemically purifying, whereas exercise seems to be reducing the endocytic rate from a constitutively recycling compartment. So the constitutively recycling compartment and the specialized compartment or storage compartment, they share many proteins, but not all of them. And in particular, there's work actually from Ralston and Galbo many years ago showing that the transferrin receptor is a marker more of the exercise pathway. Final point, in purification studies, Andre Moret and us have shown that indeed there's more transferrin receptor with GLUT4 in the recycling compartment that responds to exercise. So I think all of that points to different traffic modalities drawing from different.
Thanks, Dr. Klipp. Leaky will hold all questions unless they're Roe GTBAs related for now. Thank you so much for a wonderful, wonderful presentation. I have to hold your attention for just a minute. Greg, I'm sorry we're a little behind schedule, but we are not leaving this room until you're completely done, don't worry, on the shoulders of a coffee break. Dr. Cartee is a professor of movement science, associate dean for research, and director of the muscle biology laboratory at the University of Michigan School of Kinesiology. He's a true giant in the field of muscle physiology and glucose metabolism. He's honored with the ACSM Citation Award, which is the highest honor that the organization gives. He did his PhD in Texas, correct?
Yes.
And then did postdoctoral work in John Holosi's lab during what I would call super productive years with 12 publications with John and 1 postdoc, which is incredible. You are really a gentleman and a hero in my eyes, and we look forward to your talk today on the role of AS160 expression in skeletal muscle glucose uptake. Thank you.
Great. Thanks, Mike. I appreciate it. I want to thank the organizers and especially David for inviting me. And it's been a really interesting session so far, so I'll try to not let you down. But this is a special meeting, the IBEC, whenever it's the usual IBEC, but of course this year is especially doubly so. I also mentioned that at Toronto, when I was a postdoc with John Holosy, I came up here for about a month and worked in Amira Klipp's lab. She was very generous, and so it's kind of nice to be back in Toronto. So, and I always want to start off with my first slide to acknowledge the people that did the work, to make sure that you realize that and don't get confused that I did the work. I'm especially focusing on Ed Arias, who's been with me for more than 20 years. Amy Xing, Haiyan Wang, and Song Un Kwok, who has a poster here today. It's today and tomorrow. I would encourage you to go see that. It's related to glucose transport, but really it's working out some methods to look at something a little different than what I'm talking about, going back to a molecule glycogen that people have been trying to understand the relationship to insulin sensitivity for years. It makes a lot of sense that it would be related, but it's been very difficult to isolate. So please stop by there. I'll mention my collaborators when we get to that point, and thank you to NIH for the funding. So I don't think I think this is a rhetorical question to this audience. I think you know the answer. Indeed, exercise does increase insulin sensitivity, and we'll talk a little bit about that. Thank you, Licky, for not stealing all of my thunder. So this is actually also important, I think, to say it's 40 years since Eric Richter did this seminal study where he really rigorously showed for the first time that exercise improves insulin sensitivity in skeletal muscle. People had suspected this for a long time, and there was evidence around that, but he did this beautiful study. So it's showing in acute exercise, which is going to be what I'm going to focus on, is 1 bout of exercise. And the groups here were sedentary and exercised animals. It's a treadmill running protocol. He used a perfused hind limb, which is mostly going to be skeletal muscle that's taking up the glucose uptake there on the y-axis. And you can see on the x-axis he had a full dose response with insulin. And you can see with the open circles that there's, as anticipated, insulin-stimulated glucose uptake until it levels off. And then what's really clearly evident is that with one bout of exercise, the triangles, there's a leftward shift to the dose response curve. So this is a classic improvement in insulin sensitivity. And so you can basically see the half maximal insulin concentration for glucose uptake in the sedentary animals. And then, whoops. And then that's moved to the left in the exercised. So this was actually, as I said, 40 years ago. It was actually, I'm afraid to say, not too many years later than that I started studying this. So in 1985, I began working in this area, working with John Hollisey, and I still am trying to figure it out. Okay, so this is a slide that shows, when we think about in a physiological context, glucose uptake in the muscle that's going to be regulated at a number of different steps. And obviously during exercise, as Leakey said, blood flow is going to go up quite a bit in the contracting muscle, and so that's going to be really important for glucose delivery or supply. But for the experiments that we're looking at, it's really post-exercise. We're looking several hours after exercise when blood flow is reduced. Of course, it is true that insulin can increase capillary recruitment, so that can play a role. So supply can still play a role in this phenomenon, but we and others have used isolated muscles where we see a really robust increase in insulin-stimulated uptake. So there's really things that are intrinsic to the active skeletal muscle. We've been talking about glucose transport and GLUT4. So again, GLUT4, as Mark Hargrove said, can, with acute exercise, you can increase mRNA expression, but you typically don't have large changes in protein abundance, at least under the exercise conditions that we're studying. So what I'm going to be talking about is not GLUT4 expression, but it's changes in redistribution of GLUT4 to the cell surface membranes. And there's evidence, it's clear that exercise can improve the amount of GLUT4 in the cell surface membranes in response to insulin. And we and others have used 3-O-methyl-D-glucose, which is a non-metabolizable analog, which accumulates at a much higher rate after exercise. So that's clearly a transport phenomenon as opposed to metabolism. Now, that's not to say that metabolism can't modulate these effects. And so that's kind of, again, for Songun Kwok's poster is looking at glycogen, not so much for the stimulation of glucose uptake, but for the reversal of the effect of exercise. Okay, so when we look at exercise and the effects on insulin signaling, when I first started this in the 1980s, we pretty much knew insulin bound to the receptor, and then a miracle occurred and glucose went into the muscle. So obviously through the years, we've learned a lot about the complicated insulin signaling pathway, and when we talk about acute exercise, most studies really have not seen an increase in proximal signaling ranging from the insulin receptor binding to the activation of AKT. There are a few exceptions, but most studies have not seen this. So it seems to be something post-AKT, which we've known for a long time is really important for regulating the GLUT4 vesicle translocation, but there was a time when we didn't know really anything about what was the intermediate step. So in 2002, Gus Lienhardt at Dartmouth identified a protein that he named AS160, or actually AKT substrate of 160 kilodaltons, or AS160. And the really more formal name is TBC1D4. So this AS160 is an AKT substrate. It's a Rab GDPase activating protein. So under the basal conditions, in the absence of insulin, it tends to favor Rab GTP hydrolysis. So you tend to have more Rab GDP, which favors the restraint of the GLUT4 storage vesicle. When you phosphorylate on key AKT sites, with the most important likely being threonine 642, and the second most important probably serine 588, you can actually release this restraint. So this is one way of saying this in a more simple way is unphosphorylated AS160 appears to serve as a brake that inhibits GLUT4 exocytosis, and phosphorylation seems to release the brake. So again, now that we know that AS160 links AKT to GLUT4, the obvious question was, does exercise alter AS160 phosphorylation? So we did some experiments a number of years ago, and I'm just going to summarize that. But before I do that, The experiments I'm going to be presenting are all in the isolated rat epitrochlearis muscle. This is a forelimb muscle that has a number of features that makes it very useful for our experiments, one of which is very thin. It's about 20 fibers thick. It's actually translucent, and so when you put it ex vivo, it gets nutrients and oxygen quite efficiently. It also seems to have a fiber, or does have a fiber type composition quite similar to the average fiber type composition of a rat. And it has some other features that make it useful. So the typical experimental design that we're doing is an in vivo exercise protocol. We're using a protocol that we sort of modified slightly from what John Hollisey used for years. And it's 4 times 30 minutes exercise. So the epitrochlearis is recruited very effectively by this protocol. And we typically use something similar to this. And for the studies that I'm going to be talking about today, we wait 3 hours, 3 to 4 hours post-exercise to isolate the muscles. Because the rat, of course, has 2 epitrochlearis muscles, it's nice you can incubate one muscle with insulin, one without insulin. So you have a nice ability to see the differential effect of exercise with and without insulin. And then in the final step, we include a radio-labeled analog, usually either 3-methylglucose or 2-deoxyglucose. So again, returning to the AS160 structure, and just again highlighting there's actually multiple AKT phosphomotifs on AS160, but these 2 sites that I mentioned before seem to be the most important for GLUT4 regulation. So just to summarize what I've said so far, it's very clear exercise increases insulin-stimulated glucose uptake, acute exercise. This seems to be largely attributable to greater cell surface GLUT4. Most studies haven't seen with acute exercise proximal signaling steps being altered, but we have seen greater phosphorylation on one or both of these sites, and threonine 642 and serine 588. And so this is just 10 different studies that we've done this, so it seems to be quite a robust observation that we see. And again, we've used this mostly in rats. We have done some work with mice, but in our hands, we can get an increase in insulin sensitivity in mice, but it's not as big of an effect, and it's a little bit more variable. And I understand other people have seen the same thing, so we've really stayed with rats, largely for that reason. So at this point, really, circumstantial evidence points towards the hypothesis that AS160 may be important for this increased insulin-stimulated glucose uptake. But there really hasn't been direct evidence to support this. So as I said, because we're really interested in, I said that the exercise works in our hands much better in the rats. Even though there were AS160 knockout mice available, we went to the trouble to make an AS160 knockout rat, because we thought ultimately we're physiologists and we actually want to study the function at the end, and then we need to really have a good functional outcome. So we were fortunate to have Tom Saunders, who's the head of the Transgenic Animal Corps at the University of Michigan, to help us with making the rats, and Nathan Chi, who runs the Metabolic Phenotyping Corps, who helped us with some in vivo phenotyping. So what we saw is these knockout rats, which are indicated by the black circles, were in fact glucose intolerant. They were not diabetic, but they were glucose intolerant, and this looks a lot like the knockout mice that had been created by several different groups. They were insulin resistant, not surprisingly. This also corresponded with what had been seen in the knockout mice. So the open symbols are the wild type, and the filled symbols are the knockouts. And we also looked at glucose uptake in vivo, which we saw insulin resistance in the muscle, as you would predict. But because we were going to use an isolated muscle model, we looked at the insulin resistance here. And so you can see the minus plus insulin, the first 2 bars are the wild type rats, and the second 2 bars are the knockout rats. So if we really focus on the black bars, you can see that they're substantially insulin resistant, and this is in the epitrochlearis muscle. Fortunately, they didn't have any AS160. That's what you want to see when you have a knockout rat. And so there was no AS160, either total or phosphorylated. But also like the knockout mice, the proximal signaling at AKT seemed normal. But it is important, one thing to keep in mind with these animals, they actually do have lower total GLUT4 abundance. And again, this has been seen in each of the mouse knockouts of AS160. It's not because of a change in the amount of GLUT4 mRNA, but it's a change in the lysosomal degradation of the protein, likely because you're not restraining the vesicle and holding it in. It's probably cycling at a high rate, and that might increase the odds of being degraded. So the first simple question was, is AS160 essential for the increase in insulin sensitivity after exercise in skeletal muscle? And so this is just the wild-type controls. It's important to say for each of these experiments, we have contemporary controls. These are not repeated experiments. It's a separate experiment. So this is wild-type rats, and if you focus again on the second and fourth bar, you can see a really substantial increase in insulin-stimulated uptake, which is quite fully expected. What we saw with the knockout rats is there is an exercise effect, both with and without added insulin. But you can see it doesn't really look like insulin's an enhancement in the exercise muscles. And so to really look at this more specifically, we calculated delta glucose uptake. So remember, these are paired muscles from the same animal. So we subtracted the glucose uptake without insulin from the glucose uptake with insulin for each pair. So now we can look at the two genotypes together, and so you can see the expected increase in insulin-stimulated glucose uptake, the first two bars, in the wild-type rats, but in the knockouts, there's really no evidence of increased insulin-stimulated uptake. So basically, in this model, AS160 is essential for this increase insulin-stimulated glucose uptake post-exercise. Okay, so of course, though, what we need to do is see if we can really rescue this by putting AS160 back into the muscle to really be convinced of what we're looking at. So we use an AAV approach to restore AS160 expression. So we collaborated with Dongxin Duan and his colleague, Dr. Pan, who are at the University of Missouri, who are gene therapy experts, muscle gene therapy experts. So basically, just to go through this just in a little bit of detail, so we had wild type and knockout rats, the two muscles, so we anesthetized, surgically exposed the epitrochleus. It's so thin that if you tried to just inject it through the skin, like you might do in some other muscles, that would be a problem. So we exposed the muscle, inject the AAV vector. So we used a control alkaline phosphatase for the wild-type rats and delivered wild-type AS160 into the knockouts, sutured close, and came back 3 weeks later and performed the exercise. So we were successful. Obviously, that's what we had to do, is we got the AS160 back to not statistically different and certainly not lower than what we had in the knockout rats and in the wild-type rats. Again, this is a separate set of animals, but you can see, once again, increased insulin-stimulated glucose uptake in the control wild-type rats. And in fact, there was a significant increase in the insulin-stimulated muscles in the knockout rats that had rescued the AS160. And when we calculated the delta insulin for glucose uptake, it's not different between the two. So in other words, we were successful in restoring AS160, and that restored the insulin-stimulated glucose uptake. As I mentioned earlier, though, GLUT4 is reduced in the knockout rats, and so that's a problem, right? Because the GLUT4 is essential for this increased insulin-stimulated glucose uptake, and that could be confounding the interpretation. So we wanted to know if we could restore the GLUT4 in the knockout rats, but not restore AS160. Would that solve the problem? Is the AS160 phenomenon really just an indirect effect related to GLUT4 abundance? So a similar experiment, except in this time, and what we did is we used PBS for our injection controls in the wild-type rats, and we used GLUT4 in the knockout rats. So again, not restoring AS160, just GLUT4. So remember, this is just documenting that we checked they, in fact, don't have any AS160, the knockout rats, but we did succeed in getting GLUT4 back to the wild-type levels. And again, this is yet another control group, but see basically the same result. We have increased insulin-stimulated glucose uptake, Interesting here, not something that we actually necessarily expected. You can see that there is an exercise effect on glucose uptake in the knockout rats with GLUT4, but it doesn't really look like there's an insulin effect. So there's a residual effect that appears on the insulin-independent glucose uptake. We think, we're not quite sure what this is exactly. We imagine, even though it's the sort of expectation is that AS160 is regulating GLUT4 exocytosis. It's possible under these conditions it also affects the endocytosis, or it's essential that it's there through some process, but that's totally speculative. But when we calculated delta insulin, it was quite clear that we did not have any insulin-stimulated glucose uptake. So restoring GLUT4 did not restore the post-exercise increase in insulin-stimulated glucose uptake. So returning back to the phosphorylation, this is really where we were trying to go all along, is the phosphorylation, but we had to march through these other experiments first. If you remember the two key sites that are AKT regulated and insulin dependent. Another phosphorylation site I haven't mentioned is serine 704, which is an AMPK phosphomotif. And exercise does increase often this phosphorylation on this site. It doesn't seem to directly regulate glucose uptake, but there's some evidence that it might have an indirect effect. So when we started out this, we decided, you could do a lot of individual phosphomutants, or we can do one to begin with, all of them, and phosphorylate, mutate all of them at once, and that's what we chose to do. So we wanted to ask if is phosphorylation on these sites essential for the increased insulin-stimulated uptake. So again, we used AAV, and so we made a triple mutant that we call the 3P mutant, where there's alanine substitution for each of these key amino acids. This time it's just knockout rats, so there's not wild-type rats. So we have contralateral muscle, one with wild-type AS160, one with a 3P mutant. So again, we've sort of shown that we can rescue the increased insulin-stimulated uptake if we put wild-type AS160 into the knockout rat. And they had AS160 phosphorylation, as you would expect. It's wild-type AS160. Interesting, we did see an exercise effect on glucose uptake in the insulin-treated muscles. So that was quite interesting. Even we did document that they, in fact, cannot there's no phosphorylation on any of these three sites. When you actually calculate the delta insulin here, there is a significant increase in the muscles from knockout rats with 3P, but it's significantly lower than the animals that got the wild-type AS160. It's about 44% lower. So it's a partial rescue, but not a complete rescue. So basically, it appears that on one or more of these sites, we can't say at this point for sure, is responsible or required for part, but not all, of the increase in insulin-stimulated glucose uptake. So I've gone through a series of experiments. I'll just go through real quickly the summary. AS160 expression is essential for the post-exercise increase in insulin-stimulated glucose uptake under our conditions. Restoring GLUT4 abundance did not rescue that effect in the knockout rats. A substantial portion, but not all of the portion, is related to phosphorylation of AS160. But the remainder is independent of phosphorylation on these three key sites. So that raises a lot of possibilities. Is it other aspects of AS160? Is it AS160 independent? We can't say at this point. Obviously, we're very interested in that. But these are the first results for a direct role of AS160 for increased insulin-stimulated glucose uptake. So thank you very much. I'd be happy to answer any questions.
Greg, that was super impressive. I'm convinced now that AS160 is something to be looked at in further detail for both type 2 and maybe type 1 to get a handle on. Is it linked to the glycogen restoration after exercise or not at all in your hands?
Well, I can say this, that the reduction in glycogen wasn't different between the knockout rats and the wild-type rats. And also, AMPK activation wasn't different between the two. We have not looked at the glycogen restoration, so that we didn't do any kind of try to recover it. We haven't tried to reverse the effect, for example, by feeding carbohydrate to these animals. I suspect that they're going to be a slower rate of glycogen reversal, but we haven't done anything with that.
I've got lots more questions, but Vicky, go ahead.
Thank you very much for a very nice talk. Okay. This sustained phosphorylation of AS160 that is seen after exercise, and that is probably, at least in part, based on your results, mediating this enhanced insulin sensitivity. So this sustainment of the phosphorylation, do you know what that is caused by? Like, do you know about phosphatases or how they are regulated post-exercise? Or
Yeah.
what do you think is the mechanism for that?
Yeah, so phosphatases, people ask us that, and there's a reason a lot of people don't study phosphatases, is they're really hard to work with. There's not as many reagents. But we did dip our toe in the water for that a little bit. And we first tried to figure out which phosphatases were relevant for these sites. And so it seemed to be phosphatase PP1 alpha was at least involved in some cell culture work that we did, so it looked like that was relevant. We have a little bit of evidence suggesting there could be a reduced rate of dephosphorylation, but it's quite indirect evidence for that. We don't have any kind of genetic experiments or anything like that. So I wouldn't bet on that yet, but there could be a role, but we also think there could be other mechanisms for sure.
Thanks.
Yeah.
Thank you. I was wondering whether you looked into the downstream of TBC1D4 and the RAP proteins. So whether we are closer to understanding, because now there's several genetic models on D1 and D4 and so forth, but whether we are closer to understanding what's going on after these RAP gap proteins.
Yeah, so that's a great question. The short answer is no, we haven't done it. Certainly an outstanding question. And we simply haven't taken that path. We're still trying to figure out the sort of where we are with the AS160. So I have thought about trying to do that. I feel like we have enough questions with this partial effect that right now that's probably not going to be us. So if somebody else is interested, that'd be terrific. But I can't answer the question.
Thank you.
Another quick question, if you don't mind. Studies suggest that the duration of heightened insulin sensitivity after exercise might be influenced by the duration of the activity and the intensity of activity. And so have you thought about how AS160 could be influenced by the load of exercise or duration? Is it going to be an on-off phenomenon that's really regulating all that?
Yes, so I mean, we haven't really tried to, even this goes back a long way, going back to my postdoc in 1980s, but even before that, this may or may not be related to AS160, but glycogen is sort of a logical that it might be involved in reversing it. In other words, it makes sense that you're increasing insulin sensitivity to restore your glycogen, but we all know that that doesn't go forever. It stops, right? You might get a supercompensation. But so it doesn't quite answer your question, but glycogen is so hard to get at. People have been studying, Claude Bernard, I think he might have studied glycogen, so people have been studying glycogen forever, but to manipulate glycogen, you have to do very complicated things that are hard to interpret. So we're trying to get at that. And when we do that, if we're able to go look at Songon's poster, if we can manipulate glycogen, we can look at AS160. Yeah.
Thank you. Dr. Zurath.
Greg, thanks. That was really elegant work. And you used this 3P mutant. Is there another site that you're currently working on today?
We have, there's a lot of different ways to go with this. We're looking at 704 just because it's, as you know, potentially a priming site for the others, so we're working on that. But there may very well be other sites. Part of the problem is there aren't good antibodies for many of the other sites. So that is definitely a question, but I also think it's quite possible that it's either not AS160 or it's some other aspect of AS160. There's calbodulin binding domains, phosphotyrosine binding domains. There's a lot of other parts. So we haven't started, other than the 704 is the only one we've started to so far.
Great talk, Greg. Just related to the question about glycogen, whether it's glycogen degradation that is driving the increased insulin sensitivity. So Jan Wojciechowski in our group recently pulled data from 120 clamps that we have done over the last 25 or 30 years and correlated it with the breakdown of glycogen during the actual exercise period. And surprisingly enough, there was no correlation at all between the breakdown of glycogen during exercise and the increased insulin sensitivity post-exercise.
Yeah.
So.
Yeah, so I think what's interesting, though, with glycogen, it's really more than one thing. And what reverses the effect of insulin sensitivity doesn't have to be exactly what started it. So the reversal could be by a different mechanism, and it might have to do, in other words, it makes sense that if you, when glycogen resynthesis, we know that they correspond, that when glycogen is restored, you typically do reverse the effect, and it makes sense teleologically, even if that's not good enough. Yeah. So I think that's interesting. I am interested in the activation with the glycogen. I'm more interested in the reversal of the effect, which comes back to Mike's effect, which could have to do with how long people are highly insulin sensitive for both good reasons and potentially complicated reasons. But I appreciate that. I look forward to seeing that published. That'd be great.
Good, thanks.
Thanks so much, Greg. Incredible contributions. And thanks everybody for staying a bit late. Go enjoy a coffee, some posters, and we'll see you soon.